Really appreciate you. I'm just so touched that you're all here today. Charles, I'm not quite sure how to follow that, but thanks so much. I think a lot of oncology today, keeping in mind the stupidest cancer cell is cleverer than the cleverest oncologist, is a bit like this Japanese picture, which isn't called the river, it's not called the house, it's not called the tree. It's called paired swallows up there. A lot of oncology, a lot of science in general, a lot of dealing with patients, a lot of talking to people. It's not about what you can see. It's often about what's difficult to see or what you can't see. But I'm not here today because of any of the great successes that Charles has just alluded to. I'm here today because of this, which is sobering. And while we've made tremendous impact in treating viruses and bacteria, we've made tremendous strides in cardiovascular disease. If you look at the difference in the rates of mortality in the last 60 years in cancer, you actually can't even begin to compare them. And we all see cancer statistics in the newspapers, and any statisticians will tell you how to look at data. But to me, this is an utterly unacceptable statistic. Jax comes in. Now, why is that? Why is that statistic so bad? Well, there's a number of reasons for that. Cancer is heterogeneous, which means that evolution within a cancer selects out the most bad cells. We call that clonal selection. It can evade the immune system. It can remain hidden for many years. So if our treatments don't kill every single remaining cell, what we see in the clinic is the phenomenon of cancer dormancy, where we think we've cured someone, and then 5 or even 10 years later, or even longer, the cancer can recur. Maybe because some cells have been left, and like compound interest, 2 leads to 4 leads to 8, and so on, or maybe because a cancer stem cell hasn't been eradicated. Another reason is that cancer is a disease of age. There's no doubt about that. Cancer in children, although it pulls on our heartstrings and so forth, is actually very rare. Beyond our childbearing years, beyond the ages of 40 and 50, evolution doesn't care about our genomes anymore. Evolution doesn't care if we die. So evolution doesn't protect us beyond the age of 50 years old from getting cancer. And the final issue is that our treatments just aren't good enough. So it leads us to ask, why can't cancer be like an infection where you can identify an organism, work out what are the best antibiotics, and treat people accordingly? Because the current situation is a patient goes to their doctor with a mass, with a lump, and they might have symptoms from that lump. And here, our cancer vocabulary is limited. The patient might say, I'm tired, or I'm anorexic, or I'm jaundiced, or I feel fatigued. And the doctor takes the biopsy, and the biopsy is sent to a pathologist, and the pathologist sees a normal cell, and then the pathologist sees a cancer cell. And the pathologist looks at the organ it's from, and the pathologist says, that's a pancreatic cancer, which this shows in this CT scan. And that can then be chopped out, and the pathologist tells us about the histology, what we can see under the microscope. But that hasn't changed for the last 160 years since organ specificity was first defined in France. I wouldn't see a problem on that wall and say it's a wall problem or a ceiling problem. So my dream and my hope is that in 10 years' time, we won't say someone's got a lung cancer or a pancreatic cancer or a brain tumor. They'll go to a HER2 amplified clinic or an EGFR activated clinic or a KRAS mutated clinic. Because the reality is, we understand what causes that cancer that I just showed. We know the 3 horsemen of the apocalypse in pancreatic cancer, for example, are KRAS, P53, and c-Myc. We understand that, and we're obsessed with understanding it. But what we're not obsessed with is controlling it, and here's LMTK3, which is an oncogene that we discovered that we've added to that list. Now, because of that limited cancer vocabulary and the way we study cancer, we know that if a patient has a mutated KRAS status, We know that a set of treatments won't work, but we can't tell if a treatment will work. So we're at the stage where we can predict resistance, but we can't predict response. So how, going forwards, are we going to be able to predict response? Well, we do CT scans on patients, and again, we say this patient's got a liver cancer, and this is the liver on a CT scan, and look how clever we are, because the lumps are getting smaller. Similarly, if the lumps were getting bigger, if it was going DCBA, we'd say we weren't successful, but we don't know if our treatment stopped it going from 3 to 10 centimeters as opposed to 3 to 4 centimeters. And we just stop that treatment automatically. But we don't understand what's going on inside the signaling of the cancer cell, so we can control it, a bit like diabetes, where you can measure blood glucose 3 times a day and then treat the patient accordingly. So as Charles alluded to, my personal journey took me from these beautiful Wren buildings to Johns Hopkins in the footsteps of William Osler, who's really the father of modern medicine. And I actually got a card from the current chairman of medicine from Johns Hopkins this morning, which I was massively touched by. And Johns Hopkins is, when you get there, it's quite a rough place, really. It's a bit like The Wire in the middle of Baltimore. And you've got a concentration of people, like we have at Imperial, that are very, very smart and very, very giving as well. Guys, sorry to interrupt. I'm just concerned, shall we make room? And we've got seats on that side as well. I'm just keen for people to sit down, because this is like a 3-hour talk, just to explain. So, if that's all right. I'm just going to let you all sit down, so I'll make you feel really embarrassed while you're sort of... is there any way we can get... Jonathan, Naomi, there's seats on that side if you... Okay. So, and just in William Osler's words, the good physician treats the disease, the great physician treats the patient who has the disease. Now, when I went to Johns Hopkins, it was in the midst of the HIV pandemic. About 70 to 80% of people I saw had HIV, and there was a huge amount of activism. There were protests everywhere. What are we going to do with these individuals? Because they had elusive and strange diseases, the likes of which we'd never ever seen before. But there's a lot of overlap with oncology, and here we've stolen the ribbons from the HIV world into the oncology world. And I stood on the shoulders, if you like, of the two Marks, Brian and Francis Gotch. Thank you for coming. And undertook further research into this area, really trying to look at the link between HIV and oncology. And the good news about HIV now, and these are some of the papers that this great team I work with published, the great news about HIV is it's over. We're done. It's a problem of drug access. Regimens that now cost $20,000 a year, in 5 years' time, will cost $2,000 a year, and in 10 years' time, $200 a year. This virus was first described in AIDS was first described in 1981. The virus was isolated in 1983. And 30 years on, as I mentioned, it's a problem of drug access, and people who are HIV positive have a normal life expectancy, provided they get the right treatment. So how does that link to oncology? Well, we know in 1909, Paul Ehrlich won the Nobel Prize for the immune escape theory of cancer, in which he said we all develop cancers, but that the immune system clears them. So Kaposi sarcoma was the classic AIDS cancer with its histological subtypes. Now, anyone that knows me well knows I collect stamps, because I'm really, really cool. And Moritz Kaposi, who discovered this cancer, or first named it in the 1850s, created beautiful descriptions, but everyone died of it. And this is the virus in B cells. Now, I know we're going to talk about this slide for about 20 minutes, just to inspire you. But Sidney Brenner said the number of kilobases of DNA in an organism equaled the number of genes. So a human has 3 billion base pairs, 30,000 genes. We make about 100,000 proteins because of glamorous processes like splicing. Herpes viruses, such as Kaposi sarcoma-associated herpes virus, nothing to do with cold sore herpes, in case you're kind of worried, has 140 kilobases and 150 genes packed really tightly together. And what this virus has done is, in the course of evolution, it's hijacked our own genome to incorporate all these different proteins from our own genome, such as interferon regulatory factors, macrophage and inhibitory proteins, in this molecular arms race to compete with us. But at the left-hand end of KSH3 is this gene called ORF-K1, which is nothing like any other gene we've ever seen before. And when we studied it, we found that you could group K1 into 3 different types, depending on where people were from: America, Africa, or Asia, America and Europe. And we found, looking at all 289 amino acids, that it stimulated an immune response in this hypervariable region between 60 and 80 amino acids. But the amazing thing is, even though no two viruses of KSH3 are the same because of this hypervariability within those amino acids, what we found, and this is beautiful pictures of dendritic cells and B cells interacting to lead to stimulation and a KSH3-induced immune response. What we found was that the KSH3 virus stimulated an immune response against itself. Now, why would it do that? Well, ultimately, it preserves the host if it kills itself. So what happens during the cell cycle is KSH3 is produced, it stimulates an immune response to itself, which controls its number, so the human is preserved as well as the virus. Now, cancer is not quite as clever as that, even though this induces cancer. I believe cancer is a new form of speciation, species being defined as something with a different number of chromosomes. Cancer has a huge number of chromosomes. It has a shared lineage from the human host. It reproduces, but it enslaves us. It exhibits autogenous parasitology. It totally wraps us up. It has a different nutritional need to our own self, but it's behaving totally differently to us. And it's much easier to say a cancer is a new species than to say a brown dove is a new species of dove compared to a white dove. And in conjunction with Philip Savage and Steve Patterson, we worked out a way using B cells as a geographical framework and HLA molecules, your own surface immune molecules, linked to peptides from that virus to stimulate a T-cell specific immune response. So Kaposi sarcoma, and this great work done, is a beautiful model of oncogenesis. And it also taught me very early in my career that if you're going to produce good research and good medicine, you need a good statistician next to you at all times. And we produced a number of prognostic indices, and these were hugely exciting. Now, here, we've eradicated a cancer. It's over. Just like the HIV, because we've removed the underlying immunosuppression, we've reconstituted the immune system, we've attacked the HIV and the herpes virus that causes it, and we lead to a direct anti-tumor effect. But we also understand that we respond differently. Just before I said you can predict response, rather you can predict resistance, but you can't predict response. So chimpanzees, they can be affected by an animal form of HIV called SIV, and this is Bella from London Zoo. And that's the Sooty Mangabe, who was born while I was doing my research at London Zoo. But when you infect a chimpanzee with HIV, it never develops AIDS. When you infect a Sooty Mangabe, it always develops AIDS. And why is that? How can we apply that to cancer? Well, maybe we're not so different. Maybe there is substantial overlap. So beforehand, I showed you the horsemen of the apocalypse in pancreatic cancer, RAS, P53, and MYC. And we say that those 3 are undruggable, but that's like saying the moon is unwalkable or the internet is unsurfable, because when we drug the HIV polymerase or protease, we leave people who are HIV infected, but they have a normal life expectancy. But drugs we use in cancer treatment every day, such as gemcitabine, are not so different to drugs used in HIV management every day. But the drugs we use were discovered when the Luftwaffe bombed the USS John Harvey in Bari Harbor in 1943. And it blew up a bomb containing mustard gas, and all the sailors were doused in mustard gas. And a very clever US Surgeon General on board the ship called Colonel Stuart Alexander realized that the white cells of the Marines stopped dividing on day 5. So maybe he thought there's something in mustard gas that's inhibiting cell replication. And there, the first chemotherapy was born from that German attack in Bari Harbor. And that led to the first chemotherapies, which then led to the first antiretrovirals. So they're intimately related. And we've cured HIV, if you like, or left people cured within 30 years. But cancer has been around for billions of years. It exists in a reprimitivized state as a different species, and it's adapted over billions of years in a hostile environment. A pancreatic cancer is bathed in bile acids. A skin cancer is exposed to radiation from the sun all the time. And how do we treat it? Well, we treat it with noxious chemicals and radiation, exactly the environment it's been adapting to for the last billions of years. Now, when we talk about antigen presentation, that's foreign material presentation, we know that foreign material outside the cell is presented in conjunction with MHC class II molecules to T helper cells. An antigen from inside the cell is presented in conjunction with MHC class I molecules to cytotoxic T cells. And these pathways are faithful, but you can get exogenous antigen into the classical proteasome MHC class I dependent pathway via this receptor, CD91. And the amazing thing about CD91, apart from it being useful in immunology and cancer, is we found that it had very, very high levels on cells that were present in HIV people and cancer patients who didn't progress with their disease. We call those long-term non-progressors. And so the aim here was to perform a trial to look at a new targeted therapies. Now, we have targeted therapies in cancer. We can turn off the whole tube system, if you think of the tube system as a cancer cell. We can turn off the whole tube system with a single drug in chronic leukemia. In some breast cancers, in some lung cancers, melanomas as well. And the amazing thing is, I've just returned from ESMO in Vienna, the European Society of Medical Oncology, and you see Pfizer making this drug crizotinib, and every single pharmaceutical company is making another crizotinib, even though it's only 2 to 3% of lung cancers, just to show the lack of imagination. And they wonder why, when they make a new one, it's not as good. And so what we're trying to do, and this was taken from a recent Lancet paper that we published, what we're trying to do is change this Kaplan-Meier curve. Now, a Kaplan-Meier curve shows the survival in years against the probability of the survival. And so that could be treatment A, and that could be treatment B. But all these people would have lived anyway, and all these people would have died anyway. So the only difference you're making is here, between these lines. Now, using CD91, we extracted autologous proteins from people's own cancers in an immunology experiment in a clinical trial. And we found, over time, that there were no differences between the arms. So what we were doing, we were taking heat shock proteins out of people's own tumors, using CD91, and giving it back to them as a vaccine to stimulate an immune response against their cancers. But we saw no differences whatsoever. So we failed again. And I'm reminded of failure every day. And this is Tom, who's in the audience. And this is Ellie, who I looked after for 3 years. And she died 2 weeks before their wedding. So no one needs to remind us of failure on a general rule. And this is Pramod Srivastava, who discovered heat shock proteins. And I'm incredibly touched that he's come over here for my lecture. So moving on to other cancers, such as lymphomas and Castleman's disease, another rare tumor caused by that virus. Moving on, how does the immune system function with those? Well, we know that when you give people antiretrovirals as a model of malignancy, that cancer doesn't go away. So maybe we overestimate the immune response and underestimate cancer. And a good example of that would be vitiliginous skin. That still gets melanoma, even though we produce anti-melanocyte antibodies. And in lymphoma, you can see in the Imperial cohort that over time, and the probability of non-Hodgkin's lymphoma, if people have very good antiretroviral drugs, people don't get lymphoma. But if people have no treatment, or just nucleoside analogues, which is a very primitive antiretroviral drug, there's nearly a 20% chance of developing a lymphoma, which is very, very high. So we know that a good constituted immune system can prevent certain diseases, but what we can't say in a whole host of other tumors, such as esophageal cancer or colorectal cancer, that an immune system can, reversing that immunosuppression, can treat them. That's not going to work. Now, what can I say about these two boys? Well, Leandro, you hugely privileged to work with these guys. You have a brain the size of a planet. You understand what's going on and the fundamental mechanisms inside cells better than really anyone I've met before. George, you work harder than me. You, if you'll excuse the phrase, you have the biggest balls of any scientist I've met before. And you understand that if you don't take risks, that's a much greater risk than not taking any risks. And you're a born leader, and you don't need any more motivation, but I know how much your mother having cancer affected you. And you give the most wonderful supervision to these boys and girls. I'm sorry if you're not happy with the pictures. And we have great medical students, great PhD students. Mad, this talk is being recorded, so Hannah, I'm watching you and your career. And the other people from school who are here. Because most of this is 99% perspiration, 1% inspiration. And I myself was hugely inspired by people I worked for in Oxford, then Leeds, the Marsden, Barts. Fabulous papers. Morris, who's here, with the most amazing body of work on communication skills. Peter, your trials, and Jonathan, you've really inspired me. But a message to the audience, and a message to the audience really is that if any of you think about getting cancer, a lot of these people support Chelsea. So even if you don't believe in that, just tell your oncologist that you support Chelsea. It's just better that way for everyone. It makes everything much more simple. Now, this beautiful flower, this absolutely beautiful flower, is the human kinome, 700 proteins. And George did this monster experiment where what kinases do is they add a phosphate group onto other proteins, and that's generally a non-signal. Not always at all. Careful what I say, I've got some hardcore scientists here. But often it's a non-signal. And so what we wanted to do was remove every single one of them from cancer cells to work out what really turned on cancer cells. George, where are you? There. How many PCR reactions was this experiment?
Shall I give a number or explain it quickly?
Explain it quickly.
So we had something like 600 proteins. We had to do it in triplicates, so 1,800. Then the next round, we validated half of them, so 300 by 3. And at the last round, another 100, so around 3,000 reactions.
How long did that take?
So in order to do that, we have to do some plates. I have 96 wells, not 100, so that means 31.25 plates. And yes, I did this calculation with my phone. So it took around 2 hours, so 64 hours of work.
Did you eat or sleep?
You want me to answer this?
Yeah.
So Justin did not allow us to eat.
That's good. I'm happy.
Water, and he was giving us aspirin and antibiotics every day to keep
That's great. And who paid for the experiment?
That's the most important. It's called the kindness group, so it's 2 people, individuals that make generous donation, and it's run by Richard and Evelina Gurley.
Thank you, Richard. I really appreciate that. It's a great. The reason why really is because the grant bodies just thought it was too risky. But to me, the biggest risk is not taking risks. So what this shows, and I'm not going to go into it in detail, was knockdown of all 700 kinases using a genomic readout as a response. And we found this new kinase. Now, this is a typical protein kinase cascade, where growth factor binds to a protein on the surface of a cell and leads to activation of all these subsequent kinases. And drugs are being made against all of these. And we found this new cancer-causing gene called LMTK3. But the most amazing thing about that is we're such interesting people that we went through the autopsy specimens of about 2,000 chimpanzees because we're so interesting. And we found that none of them died of typical human cancers. They didn't get colon cancer or gastric cancer or melanoma. And if they got breast cancer, it wasn't the common form. It was triple negative. And we call triple negative breast cancers triple negative to show a lack of understanding of them. It's very rare to call something in life a description purely on the basis of negativity. But we found that this gene was new to humans from chimps, which may help explain why humans are susceptible to certain cancers that animals don't develop. And what that means is, in the chimp, there's a primitive form. But when the DNA of this gene is changed, every single change codes for an amino acid change, and we call that positive selection. So this is a totally new cancer gene that's new to humans that's positively selected between chimps and humans. And we can see in very large cohorts, and this is staining of tumors, weak, moderate, and strong, that disease-free survival, overall survival in response to endocrine therapy and chemotherapy is much better if there's weak staining, if this gene doesn't exist. So this gene clearly has something to do with the unique susceptibility of human cancers. And it induces multiple signaling pathways. And this is an array experiment that was done recently. And the point of this is we now have a huge amount of information we're being bombarded with. This experiment cost maybe $10,000. Analyzing the data from it, maybe $200,000. And we can see in these mice that if you turn LMTK3 off, which is here, the tumors are smaller. And it exists in a pathway affecting proteins, so it phosphorylates the estrogen receptor, leading to its activity. And it also inhibits this protein, protein kinase C, which prevents it phosphorylating another protein called AKT, which in turn would have led to FOXO3A degradation, but instead FOXO3A is recruited to the promoter of ESR1. And so what I'm trying to say here, it affects the DNA level and the protein level via a two-hit approach. But what's the relevance to other cancers? Because we look at 5-year survival on these Kaplan-Meier curves, which, as I mentioned before, isn't the same as cure because these cancers can relapse at any time. But really, what's the relevance to other cancers? Well, in colon cancer, in people with left-sided colon cancer, mutations in this gene seem to massively affect survival. And that is even more pronounced in females. In stomach cancer, the relationship also exists, and we're now looking to see if the relationship exists in lung cancer as well, where young, particularly Asian, non-smoking women are much more likely to have a mutation in one of their cell surface receptors. So kinases are now druggable targets. We're trying to make a drug to bind to them to block their activity. And so how are we going to do that? Well, we found that this protein, LMTK3, also adds phosphate groups to something called HSP27. And the way we found that heat shock protein 27, bringing me back to my heat shock protein work, the way we found that was we laid the protein down on this film containing over 1,000 substrates for LMTK3. But the reality is going to be much more complex than a single drug for a single pathway. And this is really the tube system in cancer. And you have to block all of those, but in collaboration with David Britton at King's, we're working out a phosphorylome in space and time. Everything phosphorylated at a particular time point under particular drug conditions. Now, what controls this? What controls the levels of these proteins? Well, we know from Watson and Crick's work that DNA is transcribed into messenger RNA, which is translated into proteins. But microRNAs can also be produced, which are much smaller pieces of RNA, and they can bind to the messenger RNA and inhibit their activity. Now, everything in biology is a feedback loop. So what Leandro's group found was that the estrogen receptor leads to microRNAs that are produced from the genome, but they then feed back and target and inhibit the activity of the estrogen receptor. So it's a perfect biological feedback loop, inhibiting it, but that's blocked and that's ruined in cancer, and that doesn't work in cancer. So a lot of microRNAs inside cells are small pieces of RNA that hybridize to the messenger RNA to tumor suppressor genes, so they function as oncogenes. Now, in some beautiful experiments that we've been doing, you can see these normal cancer cells moving, and these lines chart their growth, as you can see, in a transwell migration assay. And these are normal cancer cells growing and moving. But when we treat them with microRNAs, you can see that the cells just aren't very happy. They're just not moving very far. So we're working out the way they control cancer cell movement and control spread of cancer cells from location A to location B. Now, they're tiny genes that regulate cancer development, and they have a myriad of functions that you can use them in diagnosis, prognosis, classification of cancer type, and selection of microRNAs for therapy. And they control tumor spread as well, all the way through the process that we call epithelial mesenchymal transition, from the primary tumor to invasive growth, to intravasation and extravasation, and then metastases elsewhere, which is what we're trying to cure. We know that combined analysis between patients in London and Italy with this microRNA signature, and this is another Kaplan-Meier curve, leads to much worse cumulative survival in pancreatic cancer. And again, we're obtaining huge amounts of data. It's not a question of obtaining the data, it's a question of what we're going to do with it and how to analyze it. So these microRNAs are tiny, but they have absolutely massive functions. Now, up till now, as I said earlier in the lecture, a woman goes, or a man goes to her doctor, and the doctor takes a biopsy, and we obtain the lump, and it's sent to a pathologist who looks at it and says, you've got cancer of whatever organ it comes from. But now we can perform blood tests where we can look at circulating tumor cells. These machines can detect 1 in a billion cells in the blood, and we know that if people have more than 5 of them in a tube of blood, they have a bad prognosis. If they have less than 5, they have a good prognosis. And if the treatment takes you from more than 5 to less than 5, it's like you started with less than 5 in the first place. And the technology behind these machines is staggering. It's a liquid biopsy at a particular time point. The machine stains the cells for something called epithelial cell adhesion, for DAPI, which is a nuclear stain, and gets rid of all the red blood cells, which are anuclear. It then stains it for epithelial cell adhesion molecule. It keeps those, and it then gets rid of the ones that stain for CD45, because they're white cells. And then it stains them for a bunch of cytokeratins, and it calls that a tumor cell. But we don't know if they're living or dead. And in the famous Einstein quote, not everything that can be counted counts, and not everything that counts can be counted. But in collaboration with Katie Urch at Charing Cross, we've also recently found that at the end of life, they can help answer the question, how long have I got, doc? And we found that if people have more than 100 in a tube of blood, people live for days. But if they had less than 100, they live for many months, which helps families make plans, deciding whether to go on to other treatments, and relatives deciding, and people affected with cancer and living it, deciding where to go. So, and credit goes to Charles here, who has produced really the greatest cancer trials in Europe over the last 4 decades of his career, that we took patients with advanced cancer and circulating tumor cells that overexpressed a particular receptor that binds to a particular drug. So these are more cell surface kinases, more of these proteins on the surface of cells. And these are the names of the proteins, and these are the drugs that bind to them. Gefitinib and erlotinib in lung cancer, cetuximab in colorectal cancer, and so on. And this takes advantage of the tumor heterogeneity and the clonal biology of tumors to see if we could expand a population eligible for targeted treatment using this technology in patients affected with a large volume of disease. So the plan here was to take patients and treat them not on the basis of their primary cancer, but treat them on the basis of their circulating tumor cells. And what we found was these beautiful CTCs were often reduced in number when we gave this targeted therapy, but none of the patients responded. And I love this picture that reminds me of what we do every day. Failure, we can't spell it without you. Now, just moving on from here, I thought this lecture would be just a good brief opportunity to get up close and personal. Inaugural lectures are sort of by their nature slightly narcissistic. So I thought it would be useful if I showed you an interview that I sort of gave this week to just show a slightly other side of what we do, what's behind all the science, and just to be a bit more personal. So I was talking to Michael, who's a patron of a charity that's been set up called Action Against Cancer.
Justin, why do you choose the career path that you did?
After I was at medical school, when I was working as a junior doctor in America, I realised I most enjoyed working with very serious illnesses that affected people of all ages.
Why?
I found coughs and colds and headaches just a little bit cumbersome and trivial, although there's a lot of skill in working out which is the headache. That's the dangerous one. But it wasn't really for me, and I like things that affected people of both sexes. So combined with that and a passion for science and research, it led me naturally into oncology.
But before that, I mean, when you were a kid,
Yeah.
I mean, what was the interest there?
I know it sounds corny, but I sort of wanted to help people. And I had a lot of stories from my childhood where I saw people distressed or in trouble, and I wanted to reach out to them. And I know that's sort of the oldest thing in the book. And going through life, my uncle, who died in the forest fires with his wife a couple of years ago in Australia, was a professor of zoology there. And I used to go out and study animals with him in the bush. So, for example, one biology project I did was looking at the way these very aggressive bull ants, these prehistoric creatures, could live together in the same habitat. And we found they ate at different times in different places and different foods. And that helped me develop a very early passion for qualitative and quantitative research and trying to find out the truth, if you like, because what we think and what we believe will be the data aren't necessarily the data, and that's one reason I like research a lot, because what we think is the case is not always the case. And if what we felt was correct, we wouldn't need to do so much research.
The question you always get asked in a situation like this is, how far away are we for a cure for cancer?
Yeah. Some cancers are curable. Some cancers that, like testicular cancer or lymphoma, certain skin cancers, early kidney cancers, many breast cancers and colon cancers that we see, early lung cancers, even early brain tumours, they're completely curable, provided they can be surgically operated on. And then we can use drugs to remove what we call minimal residual disease. But a characteristic of those cancers that are curable is that you can insult the cells and those cells will die in response to genomic damage. Some cancers, though, are very good at repairing genomic damage. They're good at repairing their own DNA, and they're very difficult to cure and kill. But when I started my career 25 years ago, we cured less than that, 20 years ago. We cured about 40% of people. 10 years ago, about 50%. And now, I would say we cure about 60% of people I see walking into the clinic, which is impressive. We all love sensationalist headlines about the latest cure, and that's much more appealing than reading about some very interesting results about some cells in a dish, and people are much more attracted to it, although the cells in the dish and with interesting data are generally the reality. But good medicine, and good medicine for the future, needs to be based on a thorough understanding of fundamental, basic science, and it builds from that. You can learn from that such enormous amounts, and you can help millions of people.
What about another thing, the relationship that you have? Now, you mentioned that you like serious diseases, and of course, you will see a lot of people who have a very deep problem. They come to you, and in a sense, I mean, they regard you as being hopefully a miracle worker, in a sense. And I wondered, the question really is how would you maintain a balance, a sensible balance between your own emotions and their expectation?
Well, in terms of my emotions, I'm just very busy, so I think my coping strategy of putting my head in the ground and doing the ostrich thing sort of generally works for me, although I feel a bit sorry for my wife and friends. But I think with patients, it's generally important to under-promise and over-deliver, not the other way around. And we do. I think most patients I have and people I work with certainly don't think of me or my colleagues as miracle workers. I think they think of us as hardworking people that are reasonable to talk to, rational, and will provide choices and try our best. And when I present choices to patients, often the first choice I present is to not have any further treatment. I mean, there's 3 great quotes about dying that I like. There's the Bob Hope quote, which is, "Problem with dying is it can ruin your whole career." There's the Woody Allen quote, "I don't mind dying, I just don't want to be there when it happens." And there's the Spike Milligan one on his gravestone where it says, "I told you I was ill." But
or I'll be seeing you.
Exactly. Exactly. And so if you can make that phase of people's life as comfortable as possible, there's a phrase, friend in health, ally in sickness, companion in death. And what I mean by that is sometimes when people are very healthy, you can talk about their weather, their family, what they've seen at the cinema and theatre and so on, where they've been on holiday. When people are ill, you talk about their disease. And when people are dying, I don't think people necessarily always need rationale and reason. They just want company and companionship.
Now, you call your lecture, Curing Cancer, Overcoming Problems on the Line.
Yeah.
What does that mean?
Well, you know, I get the tube to work every day, and you can think of a cancer cell, it's an analogy I like the tube map of London. And if the drug attacks Green Park, I can still get to work by going a different route via King's Cross or a different station. It will take longer, but you can still reach that point. So if you use a very selective targeted therapy, you can often remove those important tube stations, but the cancer cell can still get there, but it will take longer. So a combined approach where you're sort of blitzing all the tube stations and then focusing on what we call the driver tube stations as well is much more likely to work. So there you're looking at combinatorial medicine, if you like.
So how did you get involved actually against cancer?
I have to thank one individual for it called Hilary Craft, who's in the audience and has 3 different cancers that have spread: leukemia, lung cancer and breast cancer. And she's done just remarkably well. It was a charity that together was set up to, you've been to the labs, you know how hard everyone there has worked, and you've met a lot of people there. It was a charity designed to help fund that work, and it's meant to have just very low overheads, very low bureaucracy, and act quickly. So we can make a discovery and then build on it immediately without waiting eons of time to get involved, basically. And it's just been wonderful. And Sam in the audience, who's got a metastatic cancer, has organised huge walks for people, Kathy's coffee mornings, and every little bit helps. And I'm just very, very lucky to be involved with people trying to ultimately help sick people get better via this charity in that way.
And what's next for you? I mean, 20 years or so in the business so far. You've got another 20 years or more, maybe.
Or less.
God willing. So what would be, for you, a satisfactory conclusion to that ambition that you started off with all those years ago?
I think people like me have a bit of an intrinsic problem that we're never that satisfied. But it would be nice to be able to look back and say, we wish we'd made a difference. And also being a reasonable father and husband and so on. Sometimes and friend, it's difficult to combine all those things, as people testify to. But it'd be great if that would be possible.
Justin, thank you so much.
Thanks, Michael. I genuinely now feel I need to lighten the atmosphere. I just think it's sometimes I feel bad for those guys in the lab. They don't get paid much. They work unbelievably hard, and they can do work for a year and not produce any results that are publishable, which is fortunately or unfortunately a measure on how we're judged. But I just implore you not to give up. And sometimes we all think about giving up from time to time, and I genuinely think that most of it is about perspiration as opposed to inspiration. In terms of going forwards, I really think, I genuinely believe we can have an end to failure. So we can put this together, for example, with LMTK3 by studying the molecular function and regulatory properties, by elucidating the effects, investigating the signaling pathways and drugging it, which I have to thank Richard for his support. And I have to thank other people too who've been incredibly generous with their time and energies and other forms of support as well. And understanding the way microRNAs interfere with all of these pathways and inhibit them. So the future of cancer care, I think, will be systems and collaborations, not individuals. Great research now is going to involve large bodies of coordinated people. Some of those papers contain 6 institutions scattered around the world, huge numbers of people. But it's worthwhile remembering that advances in knowledge is made in small steps. It's only the bit in the middle of the Kaplan-Meier curves that we're making a difference with. The people above the curves would have lived anyway, below they would have died anyway. But we're just trying to edge it up all the time. Personally, I believe that integrating individual kinase and microRNA biology with global networks with a background of immunology will be the answer, but we can only try our best. But at the same time, we can use blood tests. We now have a big project to look at circulating free DNA in conjunction with Lester and Jackie Shaw to provide real-time measures of results, and together, an end to failure. Now, there's a lot of people I need to thank. I need to thank the NIHR for funding my professorship. Thanks for coming as well. I don't know what to say about patients. They go through such hardship, and sometimes they'll complete a course of chemotherapy or radiotherapy, and at the end they'll say, well, am I cured? And the reality exists today that we just still say, I don't know. I'm incredibly grateful to the nurses and research nurses, PhD students and postdocs who humble me every day. And I'm not just saying that. I'm grateful for everyone, and Anthony at Action Against Cancer, and my friends who I think put up with more than most, as does these two rascals and my lovely wife Amy. And that's all. Thanks very much.